Let's see if this was fair. You ready? You got your copy, so you can jot little notes on. Briefly explain what herd immunity is and how it's achieved. I'm going to kind of quickly go through these and just in my words say, but as long as you had something close, you have herd immunity means that the entire population is protected because, and it's achieved by having enough people immunized. And that number needs to be well over 90% that are immunized to think that you might achieve herd immunity. It doesn't have to be 100%, but needs to be well over 90%. Name a fungus, genus, and species that's often associated with meningitis or advanced encephalitis in advanced age or other immunodeficiency disorders, or people whose immune systems have just crashed, maybe because they have cancer or whatnot. So it's not just in the advanced AIDS patients, it's in any of those categories. It's Cryptococcus neoformans. Name at least one cell structure bacteria possess that human cells don't. And what I was hoping you remembered from all of that the other day was a cell wall, right? Cell walls that have this peptidoglycan layer that's thick and gram positives, thin and gram negatives. But we can target that cell wall, and this is why you want to know, we can target that for treatment and know that that treatment won't hurt us because we don't have a cell wall, do we? Our cells don't have a cell wall, so I was hoping you put that. So define chemotherapeutic. What's a chemotherapeutic? It's any chemical that's used to treat. So it might be an antibiotic, might be an antifungal, just any chemical that's used to treat. A lot of people think about chemotherapeutics as being the drugs that are used just for cancer, but are they the only chemotherapeutics? No, anything that's a chemical that's used to treat. What's a plasmid? I wanted you to say that it is an extra piece of little of DNA that some bacteria possess. Some bacteria have little plasmids, which are just little extra bits of DNA that they possess. The way bacteria divide, what is a type of cloning, it is called prokaryotic fission. These, you would want to be answering these because, if you didn't know these, because these types of questions will be in multiple choice format on your first test next week, right? The way bacteria, what specialized cell structure is able to move a cell? They are flagella. A flagellum is singular, flagella plural. And do all cells possess these structures? No. We can actually use flagella stains to help us to try to narrow down our definitive diagnosis, right? And we know what a definitive diagnosis means rather than presumptive. So we use things like specialized stains, capsular stains, and flagellar stains. I didn't ask you about capsules, but capsules give, those capsular stains look like the bacteria have a halo around them, don't they, when there's a capsule? And it's not just that they're pretty. The capsule actually is a virulence factor. Capsules help bacteria to be able to adhere to cells that they will eventually maybe cause harm to, but also to evade or hide from immune systems, right, of the host. So these are types of special stains we use. What is the name of a specialized cell? Oh, in extreme, yeah, that's the next one. In extreme environmental stress, Clostridium bacillus, these are called endospores, aren't they? Not all bacteria can produce endospores, but Clostridium, the genus Clostridium and the genus Bacillus can. So we know this is how anthrax can live in soil for thousands of years, right? And come out later. So what is meant by the reference therapeutic index? What I kind of wanted you to put was this is an index that's used to tell us how effective the drug will be against what you're treating and how safe, and at the same time it's telling you how safe it will be for the patient, right? So we want a drug that has a really high therapeutic index, which means it's going to be safe for the patient, but at the same time probably effective against what you're targeting. Fair enough? And y'all would have said that in a much quicker way. How do you feel about this? And you got 10.3 in here, and I appreciate it. Did you feel pretty good? Did you feel pretty good? Nobody left blanks, did they? I did, okay. Well, I didn't leave blanks, but okay. Now look, this is what I'm going to do, and I'm going to pause the recording because you don't want you won't want to hear this. I'm going to be giving out something that I think you're going to be happy about. So I'll let the people who aren't here hear this, because you all will pick these up the next time you come. Y'all. Yeah. There it is. There it's all going. It's thicker at times. What did I tell you this whole class is really about? That when you hear a microorganism, you know what category it belongs to, and that's important so that you know what you're treating, how to treat, and what you're treating, but also the name of that organism brings to mind some things some diseases that organism can cause, microorganism, right? So this whole class is really that. We get to do other fun stuff, you know, or talk about other stuff, things that are going on. But that's what this class is about. And I said that your final exam would be comprehensive on any of the microbes that we've discussed during this semester. They would be fair game. Is that right? So this morning, I came in here and I thought, you know, it can be overwhelming because I see your faces sometimes when I'm talking at giving genus and species names of microbes, and I think, oh, yeah, I see your looks. So let me tell you what I did. I had, and I'm not going to promise it's complete, but I'm going to make this a living working document that you all, I myself, we're going to use this. It's almost complete. If it's not complete, it's 95% complete. Okay. I've got blue, even with punch holes. Okay. These are bacteria. You'll each get one. Now look, and you don't have to know them all now. But what I would recommend you to do is kind of go through it. And if it's one that we've already talked about, put a little asterisk or something, because you should be knowing it by now, right? So these are bacteria, blue. The green, and they got punch holes, the green are protozoa, right? Are going to be protozoa. The yellow are going to be fungi. I've left some blanks underneath because we might need to add some, okay? Please keep up with your copies because I'm not making any more. I don't know what color that is, but whatever color that is, these are helminths, which means worms, essentially, right? So that's that color. Pink are viruses, so they're not even in, you know, any of the six kingdoms or 3 domains, but these are viruses because they're very important to us, right? So pink are viruses. And then the last is actually just a little bonus extra, are going to be prions, which I haven't really talked much about, but then also animal insect vectors. Vectors like the Anopheles mosquito, which we know is what transfers Plasmodium, which is the protozoa that causes malaria. So I've got some vectors like the sandfly, the tsetse fly, Anopheles mosquito. So this is this last little whatever color that is, okay? So you all get this, and it's pretty much done. And that's going to be what you're studying for your final exam. But also a lot of questions that happen on these tests. Because like next Wednesday, am I going to be able to ask you about any of the ones we've already talked about? Right. So what I'd like you to do at the end of I'm going to hand these out, we're going to pass these out right now, but I kind of want you to put them away. But before you leave today, after we finish looking at these protozoa, the living protozoa in the lab, before you leave today, or certainly but let's say Monday, before you leave Monday, I'm going to have a consensus among you all and check on my copies, which ones y'all say I've already talked about. I know which ones I think we've talked about, but by the end of Monday, I'm going to get what you all say we talked about. And would any of the ones we've talked about today be fair to have a question about on this test next Wednesday? Just a multiple choice question, like Cryptococcus neoformans belongs to which category? And you pick fungals, right? Fungus, right? So, which belongs to the fungi kingdom. So, fair? Mm-hmm. All right, so this is what we're going to do. And let's start handing them out. So you have to get one from each of the colors. Blue is coming around now, and actually there's an area that it's stable. Okay, so get one. Please only take one because I'm going to have a few extra because some people are out. Get one of the blue. Y'all can start passing those around. Get one of the green. Are y'all happy? Well, you might not be too happy when you're seeing how many it looks like, but it's not going to look like many by the end of the semester. By the end of the semester, we're going to have talked about them so often. You know what I mean? They're going to just seem like your friends. They really are. Get one of the yellows. Get one of the yellows. Pass the yellows. Thank you. Is that extra blues? Thank you. I do need any extras up here. Get one of this, whatever color this is. Sorry, I knew this would take a little minute, but anyway, get the pinks. The pinks are the viruses, right? Sorry, Andy, you might just want to stand up. Pinks coming around. And then there's this, what is this? Buff color that's on that. All right, and so we're going to figure out how many of each y'all should, oh, sorry. We're going to figure out how many of each y'all should have, okay? I mean, you should only have one of each. But anyway, how many pages are there? There's 6? Are there 6? Really? Okay. Perfect. Thank you. Well, I figured that this, instead of you all creating your own document and me not knowing for sure if y'all really had what you needed, you know, this is just better. Right? I have not done this before, so you all are new to this. Does everybody have one that starts with prions? It's this color. Does everybody have one that's pink and starts with viruses? And it's back and front. Everybody has that? Right? Does everybody have one that starts with helminths? Helminths. Does everybody have one that starts with fungi? And everybody has one that starts with protozoa? And everybody has one that starts with bacteria. Is that right? All right. Cool. Great. All right. Now, what? There is spelunker's disease, and it's histoplasmosis. It's scary, but we haven't gotten to it yet. So, but we will be talking about it. And it is referred to as spelunker's disease. It's very common. I worked with a 6-year-old on a case where a 6-year-old from Southwest West Virginia died from that. So usually people won't die from it, but there is, you know, it's pretty serious. But anyway, we'll talk about it. Look, so I didn't turn the recorder off, so everybody that's not here got to hear me passing out papers. You all need to make sure you get your copies, those of you who aren't here. All right, so you all have those. Before we leave Monday, I'm gonna need you all to all kind of agree on which ones we've talked about, just so you all know what to review for the test that's coming up Wednesday. And then as we go through, I thought this was a perfect time to give it before the first test, because as we go through, we'll be checking off more, right? And any of the ones we've ever talked about would always be fair game to ask questions about. Okay, so look, this information today, I'm only gonna lecture for about 10 or 15 minutes probably. Hold me to it. Because then we're gonna go to the lab, and I do want some people, for everybody to get to look at these So eukaryotes, what do you know? That these are going to be those 4 kingdoms: protists, fungi, plants, and animals, right? We already know that these are going to be more complicated cells because they have true nuclei, don't they? So eukaryotes, true, means true, no matter where you see that prefix, it means true. Karyon means nucleus. So they have these true nucleuses, but they don't just have a true nucleus. They also have very complex organelles that have very specialized functions. Almost everybody in here, I know, because I've had most of you, already have been introduced to many of these organelles, but I do want to just remind people what they do, some of these organelles, the special functions of what they do. And so I will go through that and make sure I say that out loud. We know that the first life on this planet, or you should know, actually was simple single-celled organisms, but how these eukaryotes actually evolved and understanding how they evolved is that they did have these common ancestors, if you will, and probably 2 prokaryotes started living in synergy, and this is how eukaryotes actually evolved. That whole idea about how eukaryotes evolved, you can learn more about it if you want to, but it's explained in the theory of endosymbiosis. And I do think that people need to just know that scientific theory is called the theory of endosymbiosis. That's one word, endosymbiosis. And it's a scientific theory. That statement explains how complex cellular life, eukaryotes, evolved from prokaryotic organisms. We know that the archaea, or we also refer to this as archaeobacteria, are what's present today on this planet as far as eukaryotic life. We came from archaea, bacteria. So that is also what is being supported in the scientific community. So much of this we've already talked about, haven't we? In the eukaryotes, the single-celled organisms belong to that protist group, are all single-cell. And then when we talk about fungi, there are a few single-cell fungi, but most of the fungi and helminths that belong to the animal kingdom, they're multicellular organisms. Yes, Dakota, you had a question.
Can you repeat endosymbiosis?
The theory of endosymbiosis, it is a scientific theory, just like the cell theory and theory of evolution, which means, is it just some idea out there, or has there been, through all the experts in the field, have there been kind of exhaustive scientific methods being used to explain this naturally occurring phenomenon, right? So it's a scientific method that explains, it's a scientific statement theory that explains how complex life, i.e. eukaryotes, evolved on this planet. Does that make sense? How they evolved on this planet. Now, let me say this to you guys, because I think you all know this. Nobody was back on the planet 3.8 billion years ago. There was nobody here to record it. Okay, so some of the things that have been done, and it's really a lot has been done as far as researching, but it's been limited in what can be done. But some of the simulated what the primordial atmospheres and the Earth looked like 3.8 billion years ago. We do know what that was, and that's kind of not even arguable. We know that it was harsh. We know that most of the life that's living today on this planet, if we somehow got transported back 3.8 billion years ago, we wouldn't last a millisecond. It was harsh. So some of the things that have supported how single-cell life came about on this planet, Miller and Fox, and it's a bunch of scientists have done all kinds of simulations, and repeated it really, have tried to recreate what we know the earth and the seas look like, and the atmospheres. And what they see forming are what are called protocells. Protocells will form and that, which are very like prokaryotes. But actually how life came to be on this planet has not been discovered yet. Not exactly has been created. It has not. So if anybody's sitting in here waiting for me to tell you how life came about 3.8, no biologist can tell you that because we don't know yet. Nothing has done it to a point. So it's an unknown. It is a big unknown. Does that mean nobody's still trying to figure it out? Because in science, what is science? Science is a way of testing the natural world to try to figure out things, right? So people are still working on it, still trying to figure it out. And we've learned a lot. We've learned a lot about what the environment was like. We've learned a lot about how some of these early cells could have formed, what it would have taken for them to form. We've actually recreated, Miller and Fox were two of the first, recreated protocells with that same primordial soup that was in the mix at the time, as long as like lightning. And was there lightning? Sure there was, which was a stimulus for energy. We can create protocells. That's been done, simulated. But exactly how it happened, we do not know. It is an unknown at this point. Yes?
So what are exactly protocells?
So protocells are these membrane-bound cells. That's a good question. So and thank you. So, but you don't need to know this for me, so y'all can just listen if you want to. Put down the pen, lean back, so relax. But protocells, and I'm glad you're interested in it, but protocells are, again, using these, what we are recreating with what we know were primordial soup, if you will, and energy, electricity involved. You can get these membrane-bound cells that actually have the beginning of proteins and some of these macromolecules that are needed inside of cells. But have scientists figured out how to create a living cell? Or no? How it was done? No. So that question has not been answered. And it is the biggest unknown in biology, is that. And it has not been answered. But many other things have been done where we see that there are organelles like mitochondria and chloroplasts that have their own DNA. They can't live on their own right now, but it is likely that they did once upon a time. So those organelles that are now organelles were probably single-cell prokaryotes that had started to live synergistically in symbiosis along beside other simple organisms, cells, and then they became more complex, right? They became dependent on each other. But that dependency was not a bad thing. It actually allowed for the single cells, like proteins, to be able to carry out much more complex functions. So all I really want you to get from me, because we can do an entire class on this, right? An entire semester on this. But what I want to get you to hear from me is that this scientific theory is the theory that is most accepted about how we got to complex life on this planet. I think I had explained to you all that it's thought that, we know when the first single cell organisms were because we have evidence of that. We have evidence of when moss-like plants, simple plants came, how long they've been on the planet. We have evidence of when complex plant life was on the planet. We have evidence of all of that, of when they came. But how did we go from having very simple life forms to these complex eukaryotic life forms? The theory of endosymbiosis is a statement in the scientific theory that explains it. And there's a lot of evidence that had to be built in before that was accepted, right? But are there still a lot of unknowns? Absolutely. Absolutely. I feel like we're cave people, you know? we're just, there's so much actually being discovered all the time now. I mean, if we just looked at medicine, which is just a minuscule, tiny part under science, if we just looked at medicine and all that's being discovered in the medicine, in biomedical community, just the biomedical community, we understand we're cave people. In 10 years from now, y'all have chosen professions where you never get to stop learning. There's going to be so much that's always going on in your fields, and that you've got to stay learning, right? Because there's just so much that's being discovered now. Yeah.
I read in National Geographic, and it was like the world is covered mostly by water, and only 5% of like the things in the water is like the discovery.
Is that so awesome? I mean, it really is, right? So, what'd you say?
That's kind of creepy. That's what I think.
That's not creepy.
There must be some like crazy.
Okay, look, we can't be afraid. You know, knowledge is power. It's not something to be afraid of, right? So this is, we're learning all the time. But you can actually see. How many of you saw the blood moon the other night? Because was that awesome? Was that just beautiful?
It was almost too long to stay up.
It was dull.
Jake and I watched it together. But anyway, you thought I lost my mind.
I
So
was at work.
I thought it was beautiful. It's beautiful. But you can't imagine when things like that happen, how back in the day, or flooding and mudslides and things, back in the day, before there was communication, back in the day, before there were things that, you know, we could explain. El Nino. You know, El Nino is really wild. If you really just look at that, you got me off on a tangent. I'm coming back here soon. But look, I'm going to come back to life soon. But anyway, there's so much we've learned that we can take some of the fear out. And we understand how things are happening, right? And you don't, knowledge is power. And at first, you know, it's a little scary. I'll tell you that. And I tell my A&P students that medical students and nursing students at one point become the biggest hypochondriacs because they're just learning about things and signs and symptoms and diagnostic disease and you know they're hearing things and they're thinking I had that sometimes. I know I've had that. And you know, so they're become incredible hypochondriacs until they get enough information. And then it starts to calm you down a little bit. But yeah, our nature is, like I said, people don't need to take drugs. You just need to read. Because nature is mind-bending. I mean, it's really, truly mind-bending. The tectonic plates and how we understand one of the things that support how old the Earth is and other many other scientific things. Just hearing about polar north and south, just hearing about that, just hearing about the meteors that have been there, just hearing about how things have changed on our planet since our planet became, I mean, it's really kind of mind-bending. It really is. Just hearing about how exact certain level, like how far we are from the sun and our orbits and how just all of those things, how exact things have to be to have allowed for life to have happened on the planet. It's really mind-bending. Don't need drugs, you just need to read, right? Because it can blow you away. Really. Okay, I'm sounding corny, so we'll move on. Maybe I always sound corny. But anyway, so we know that protists are always one cell. So they're the simplest of the eukaryotes. But you all are about to see some that are swimming around. You're going to see how huge they are. You can see them on 10x objective, which is 100 times magnified. They're enormous. The Paramecium are under 400 magnification. They're going to just be huge. But you're going to see that these are complex, way more complex than those tiny bacteria we were seeing the other day. Some of those were really, really small, with single cells. But anyway, these are eukaryotes. Some fungi like yeast, not mold, but yeast, are going to be unicellular, but many of them are multicellular. And then we have plants and animals, which we know are multicellular, which means that we have an organism that has started, they all started with one cell, but then the cells started to divide and the cells took on specialized functions. So bacteria, simple, single cell, not very complex, don't need much from the environment as far as like what they do, but then becoming way more complex as we move up the eukaryotic kingdom. So what I'd like to do too is actually just go to this little illustration of a cell and a eukaryotic cell and looking inside at all of these organelles. How many of you remember what a nucleus does? Let's write it down real quick. What is a nucleus in a eukaryotic cell? What's its function? It's to house the cellular DNA, right? House the cellular DNA. Y'all remember that? Does anybody remember the endoplasmic reticulum, which looks like it's continuous with the nuclear membrane, but it's this membrane bound organelle that's folded. And if you took a slice, a wedge out of it, it would look like this. Does anybody remember what's made in the endoplasmic reticulum? Proteins and lipids. Proteins and lipids are where they are being made. Where does the information come to build them? Where does it come from? The information to build proteins comes from where? It comes from the nucleus, doesn't it? From the DNA. But they're being built out here in this ER, this endoplasmic reticulum. Great. Do they just stay there, those products? Do they sit right there, or do they get sent throughout the cell? And sometimes even sent outside of the cell. They get sent to be wrapped and shipped. Where do they go? The Golgi. Do you remember the Golgi? The Golgi apparatus, Golgi body, Golgi complex. I don't care as long as you know Golgi. The Golgi or Golgi, you can say it either way. But this is going to wrap and ship the products that are made inside the cell throughout the cell, and sometimes even to be excreted and sent out of the cell. Right. Does all of that take energy? All of that takes energy, and that the powerhouse of the cell is said to be the what? The mitochondria of the cell. This is where most of that power molecule is produced, and that's called ATP. Everybody remember what ATP is? Adenosine triphosphate, but ATP is the power molecule of the cell, and it's mostly produced in what organelle of the cell? What organelle? Mitochondria. Good. And that's why the mitochondria is called the powerhouse of the cell. Does all of this sound like it's taking work? Yeah, so we need a lot of energy in eukaryotic cells. Prokaryotic cells don't need that kind of energy. They're not doing all of those things. Now, there's another little organelle here that looks like it's got stacks of green coins. Can y'all see that? This is called a chloroplast, and certainly animal cells don't have chloroplast, but some eukaryotic cells have chloroplast. Chloroplasts are going to be where photosynthesis occurs. Chloroplast, where photosynthesis occurs. What are two things that you know about mitochondria and chloroplast that other organelles don't have? Those two have their own DNA. They have their own DNA. It's called mitochondrial DNA for the mitochondria and chloroplast DNA. They have their own DNA. That they have their own DNA is one of the things that supports the theory of endosymbiosis. So they have their own DNA.
It's the chloroplast that produces the sugar, right?
It's exactly. This is what, plants, we think about plants having chloroplast, but some other organisms do as well. These chloroplasts are going to be where photosynthesis occurs, which means this is where the carbohydrates, the sugars are being made. Exactly. Okay, so you guys said a flagellum does what? Moves a cell. What do cilia do? The paramecium today that you see that are going to be huge, the paramecium have these little hair-like cilia all around them. They're single-celled proteins that have cilia, and those cilia actually are helping with the movement of the paramecium. But anyway, so cilia are going to be these like little projections, and they're found in some protozoa, ciliated cells. Well, human cells can have cilia too, but no, and
I just want you to know that they're found in some protozoa, though. Seeing that something has flagella and cilia, again, is that helping us get to the ID? It really is. So, okay. Capsules, I said, do what? What do capsules help with?
Protection.
Capsules can protect the bacteria, can protect the bacterium, and capsules can help them to adhere to host other cells, and it can help them to evade things. Yeah, right. So, okay, I'm moving along. There's just all kinds of pictures here, but most of this I know. If I don't say it out loud, would you have even been responsible for it? No. But I tell you what, let's go ahead. Since this chapter, I do feel like this chapter, I'm going to be very specific in what I want you to know. Let's go ahead and go to the lab. Why don't we? Okay?